Hello students, in the last class we studied classification system as well as general characteristics of the kingdom Monera. So today I will be dealing with the shapes of bacteria and along with that we will study structure of bacteria in detail. So let's start with it. Shapes of bacteria. The name of the scientist is Cohen. Cohen recognized 4 basic forms of bacteria. Cohen recognized 4 basic forms of bacteria. So I am going to write the name of these 4 forms. That is Coccus, Bacillus, Spiral and Vibrio. So these are the 4 basic shapes of the bacteria. So let's start first with the Coccus one. The Coccus bacteria represents spherical shape. Yes, this is spherical shape. If there is only presence of one spherical bacteria, there is presence of only one, occurrence of single is there, then this type of the condition, it is known as monococcus, right? If this bacteria, it occurs in the pairs, yes, if it occurs in the pairs, then it is known as diplococcus. If it occurs in the form of chains, yes, like this, chains of coccus bacteria, if they occur in the form of the chain, then known as streptococcus. Now sometimes these bacteria, they are present in the form of the grapes. Yes, bunches, in the form of the irregular bunches like this. Yes, irregular bunches like this, giving the appearance of the grapes cluster. This type of the condition, this type of the bacteria, it is known as Staphylococcus. Now sometimes these bacteria, they can also form 3-dimensional structures. Like this formation of a cube. Yes, 3-dimensional 3D structures they are formed. This type of the bacteria is going to represent Sarcine type. So got this? Now coming to the Bacillus, Bacillus is going to represent rod-shaped bacteria. Rod-shaped, or you can say cigarette-like. Now if the rod-shaped bacteria is present singly, again, what you are going to call it as? Monobacillus. Right? Again, if they are present in the form of the pairs, now you are going to call it as diplobacillus. Forming chains. Rod-shaped bacteria forming chains. Then streptobacillus. Right? Sometimes these rod-shaped bacteria, they are present side by side like the matchsticks. This type of the arrangement of the bacteria, it is known as palisade type. Now coming to the spiral one. These bacteria, they are elongated and going to be twisted. Yes, elongated and twisted. Always going to be at the flagella. Yes, flagella is a locomotory structure present in the bacteria which helps in the movement of bacteria. So the spiral bacteria is elongated, showing twisting. The last category here, that is the Vibrio bacteria, is comma shape. Yes, comma shape bacteria. Here I am representing the flagella. So these are the 4 basic shapes which were recognized by the scientist Cohen. Among these 4 shapes, any idea which shape is the most common one? Yes, the most common shape. It is bacillus type. So let's write it down here. Among the 4, this is most common one. यहाँ पर जितनी भी शेप्स का नाम लिखा है, इसमें से एक बैक्टीरिया ऐसा है जिसके अंदर कभी भी फ्लैजिला नहीं होता. They are non-flagellated, they are non-motile, and this condition is associated with coccus. Always non-flagellatory, non-motile. The flagella is absent. Now, after looking into the shapes of the bacteria, now we will study cell structure of the bacteria in detail. So now I will be dealing with the cell structure. So here I am forming the outermost covering. Yes. So this is representing outermost covering. This outermost covering, it is known as glycocalyx. This one is glycocalyx. Now this glycocalyx, it can be of 2 types, 2 categories we will be studying here. Now sometimes this glycocalyx is in the form of loose sheath. It is thin, gelatinous. If it is in the form of loose teeth, it is thin, gelatinous. This type of the glycocalyx, it is known as slimy layer. So first category, slimy layer. Yes. What type of the condition it shows? It is in the form of loose sheath, gelatinous, forming a thin layer. But sometimes the glycocalyx is rigid. It is thick. When the glycocalyx is rigid, forming a thick layer, now it is known as capsule. So second category is capsule. Now it is rigid, forming a thick layer. Now what is the composition of the slimy layer? Slimy layer is made up of polysaccharides, carbohydrates, polysaccharides. Now the types of polysaccharides which are associated with the slimy layer, they are dextrin, and levain. Yes, these are the 3 types of the polysaccharide associated with the slimy layer. Now in the capsule, these 3 polysaccharides, they are present. Yes, the polysaccharides, they are present. But along with the polysaccharides, there is also presence of amino acid. Amino acids. The type of amino acid which is present here, that is D-glutamic acid. D represent dextrorotatory. Now, glycocalyx is a protective covering. So, surely this glycocalyx, it will protect the bacteria from the loss of water as well as loss of nutrients from the cell. Now, in the forthcoming part of the lecture, I will tell you in detail that on the basis of the staining, there is presence of two types of bacteria. Two types of the bacteria, they are distinguished on the basis of staining. That is gram-positive bacteria and gram-negative bacteria. So this we will deal in detail in the forthcoming part of the lecture, but you please keep it in the mind here. So please note it down. This capsulated condition is more common in gram-negative bacteria as compared to gram-positive one. So please write it down. Capsulated condition is more common in gram-negative bacteria. Right? Now below the glycocalyx, now there is presence of cell wall. Yes, inner to the glycocalyx there is presence of cell wall. So this is cell wall. I have discussed in the previous lecture that cell wall in the bacteria, it is made up of peptidoglycan, and the peptidoglycan I am going to be represented as Pg. So peptidoglycan composition as well as its arrangement I will be discussing in detail in the forthcoming part of the lecture. So here I am not dealing with the peptidoglycan part. Now below the cell wall there is presence of the another layer, that is cell membrane. So this one which I am forming here, this is a layer inner to the cell wall. This layer, this is known as plasma membrane. This is known as cell membrane. A true membrane, it is made up of lipid bilayer. Along with that, there is also presence of protein. So it consists of lipid bilayer. Along with that, there is presence of proteins. So around the bacterial cell, there is presence of three layers, right? Three layers are there. One, glycocalyx outermost, middle one cell wall, and the innermost is cell membrane. These three layers together which are present around the bacteria represent cell envelope. Yes? 1, 2, 3. These three layers present around the bacteria together represents cell envelope, protective covering. अब सेल मेम्ब्रेन के अंदर यहां पर जितना भी ये पार्ट जो प्रेजेंट है, दिस पार्ट ऑफ द बैक्टीरियल सेल, इट रिप्रेजेंट्स साइटोप्लाज्म. Before dealing with the cytoplasmic constituents, let's discuss some important points about the cell membrane. Now, as we have discussed previously, that in the cell membrane there is presence of phospholipids. Yes, so these are the phospholipids. The phospholipids, they are present in the form of the bilayer. Yes, so you can see there, see here, two layers are present, bilayer arrangement. Now, the components which are present in the lipid bilayer, the components which are present in the cell membrane, these components, they have the ability to show the movement. The components of the membrane, they show the movement, and this nature of the membrane, it is known as fluid nature. Yes, this nature, it is known as fluid nature. Due to this fluid nature, the membrane has the ability to cause transport of compounds. Yes, due to this fluid nature, there can be movement of the compounds inside the cell. There is movement of the compound outside the cell. So fluid nature, it helps in the transport of compounds through the membrane. Now, if the fluidity of the membrane is high, if the movement of the components present in the membrane is more, there are chances that this membrane, it will disintegrate. If the membrane will disintegrate, the cytoplasmic contents which are present here, they will ooze out of the cell. The cytoplasmic contents, they will ooze out, due to which the cell will die. So it means membrane की जो fluidity है उसको maintain रखना है एक level पर, so that the stability of the membrane is also maintained. Let's see how this stability will be maintained here. अब ये जो लिपिड्स है ये मूवमेंट शो करते हैं. इनकी मूवमेंट्स को रिस्ट्रिक्ट करने के लिए इन लिपिड्स के साथ देयर इज प्रेजेंस ऑफ कंपाउंड्स. फ्लूइडिटी को कम करने के लिए देयर इज प्रेजेंस ऑफ कंपाउंड्स along with the lipids, and these compounds, they represent hopanoids. Yes, hopanoids. So what is the function of the hopanoids? They involved in stability of the membrane. They will prevent the breakage of the membrane by controlling the fluidity. So you can call the hopanoids as membrane stabilizers. Right? In the case of the prokaryotes, the fluidity is maintained with the help of hopanoids. But in the case of the eukaryotes, the stability of the membrane is maintained with the help of hopanoids. But in the case of the eukaryotes, stability is maintained with the help of sterols. And one of the important sterol which is associated with us, that is cholesterol, right? So in the case of the eukaryotes, the stability is maintained with the help of sterol. So let's write it down. For the eukaryotes, you write this abbreviation EU. So which is the membrane stabilizer here? That is sterols. Now, in the plasma membrane, you can see these ingrowths. Yes, these ingrowths. The ingrowth of plasma membrane, they are known as mesosomes. So, I am labeling them here. These are mesosomes. Mesosomes, also known as chondroid. As I have discussed, capsulated condition is more common in gram-negative bacteria, but the mesosomes, they are more common in gram-positive bacteria. Yes, more commonly associated with gram-positive bacteria. Now the mesosomes, they can be positioned in two ways. यहां पर आपको positioning कैसे लग रही है mesosomes की? Center के अंदर. This is central mesosomes. But the mesosomes, they can be also positioned on the sides. If the mesosomes, they are positioned on the side, so they can simply be known as lateral mesosomes. Right? So two types of the mesosomes are there. Center के अंदर जो है They are central mesosome. Yes, centrally positioned. जो sides के अंदर है, they are lateral mesosomes. So now we have to see what are the functions associated with these mesosomes. Now with the central mesosome, there is attachment of the nucleoid. Nucleoid, that is the primitive nucleus present in bacteria, right? So to the mesosomes which are central, there is attachment of the primitive nucleus. We have already discussed this primitive nucleus. It is known as nucleoid, right? Now the function of the central mesosome is that they help in cell division. Yes, they help in cell division. Whenever the cell divides, all the material which is present in the cell, it undergoes duplication. So there is also going to be duplication of DNA. There is duplication of nucleoid. And this DNA duplication, there is help of mesosome is required. Before the cell divides, all the contents which are present in the cells, they are duplicated. So there is also duplication of DNA. And in this DNA duplication, in the duplication of nucleoid, there is involvement of mesosomes. Because the DNA, the nucleoid, it is attached with the central mesosome, right? So they help in DNA replication. Because the central mesosomes, they are attached to the nucleoid, they are attached to the DNA. Now let's see what is the function for the lateral ones. Now lateral ones, they contain respiratory enzymes. Yes, they have respiratory enzymes. Now which respiratory enzymes they are associated with the lateral mesosomes, that we have to see here. In the previous classes, in the 10th standard, you have studied that respiration, aerobic respiration, is mainly divided into the 3 steps. Yes, 3 main steps are there. Glycolysis, Krebs cycle, and ETS, that is electron transport system. Yes. So what are the 3 steps? Glycolysis, Krebs cycle, and ETS. So let's write this down. Three steps involved in the aerobic respiration. Glycolysis, Krebs cycle, and ETS, electron transport system. Let's see what is the positioning of these steps in the case of the prokaryotic. Write PRO for prokaryotic and eukaryotic cell. तो देखना है प्रोकैरियोटिक और यूकैरियोटिक सेल के अंदर ये जो स्टेप्स है ये कहां पर होते हैं. Let's start with the glycolysis. In the eukaryotes, the glycolysis will occur in cytoplasm, and in the prokaryotes also cytoplasm. Krebs cycle, do you remember where the Krebs cycle will occur? In eukaryotes, right? It occurs in mitochondria, in the mitochondrial matrix. But what about the prokaryotes? We have discussed in the prokaryotes the membrane bound organelles, they are absent. mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, they are absent. So this Krebs cycle, again it will occur inside the cytoplasm. Yes, cytoplasm. ETS in the eukaryotes, it will occur in the mitochondrial inner membrane. माइटोकॉन्ड्रिया के आसपास दो मेम्ब्रेन है, outer and inner, and this electron transport system, it will occur in the inner membrane. Now ETS in the prokaryotes. Whenever the ETS will occur, one of the essentiality is this presence of membrane. As in the case of the prokaryotes, the membrane bound organelles, they are absent. Only one membrane is present in the prokaryotes. Which is that membrane? That is plasma membrane, cell membrane. So surely this part of the aerobic respiration which requires membrane will occur in only one membrane which is associated with the bacteria. That is plasma membrane, cell membrane, or its ingrowth. What are those? Ingrowth, that is the mesosomes. So you got this? So in the case of the bacteria, there is presence of only one membrane. That's why the bacteria, the prokaryotic organisms, they are known as single envelope system. Presence of only one membrane. So what is the function of the mesosomes? They are going to have the enzymes for which step? ETS, electron transport system. So I'm going to write the name of the few enzymes which are the part of the ETS and associated with the mesosomes. So you please write them. One is STH, that is succinate dehydrogenase enzyme. The next one is cytochrome oxidase, right? Now STH, succinate dehydrogenase, it is an enzyme which is the part of both the electron transport system as well as Krebs cycle. Electron transport system का भी part है और Krebs cycle का भी part है. So can we say it is link between these two steps? Right? STH is the part of both Krebs as well as ETS. So in the bracket you can write it down. It is a link between the Krebs cycle and electron transport system. Right? So these structures we have discussed. What are these structures? They are the ingrowths of the plasma membrane. But can you see here, along with the ingrowths, there is also presence of these outgrowths of plasma membrane. So now we are going to deal with the outgrowths of bacterial cell structure. Outgrowths? Yes. These outgrowths of plasma membrane, they can be of two types. One is fimbriae and the another one is pili. Two types of the outgrowth of the membrane is there, fimbriae and pili. The singular of pili is pillus. Right? Now the fimbriae, they are small size, they are thin, and in a single bacterial cell, the number of fimbriae which can be present, that is ranged from 300 to 400%. Right? Pili, now they are comparatively longer. Thick. Number is 1 to 5 per cell. So these are the differences between fimbriae and pili. But please note it down, fimbriae and pili, one common function for them is they are involved in the attachment. They help in the attachment of the bacteria to the different surfaces. Second similarity is that both of them, they are made up of same type of protein, that is pillin protein. So the two similarities, one helps in attachment and second made up of the same type of the protein, that is pillin protein. Attachment and formed of pillin protein. Now, after the ingrowths and outgrowths, now you know this part is going to represent the cytoplasm. As we have discussed, in the cytoplasm there is absence of membrane bound organelles, but here there is presence of non-membranous organelles. Non-membranous organelle, what is that? That is protein factories. Yes, please tell me what is the name here? What are the protein factories? They are the ribosomes. Yes, protein factories, they are the ribosomes. And the types of ribosomes which are associated here, they are 70S type. And in the eukaryotes, you will study 80S type. Now, in the cytoplasm, there is presence of storage granules. Cytoplasm के अंदर storage bodies present है. Yes, there is presence of storage bodies. These storage bodies, they can have organic or inorganic compounds. Around the storage body, the membrane can be present, but this membrane is not a true membrane. Yes, so what is the definition of the true membrane? The definition of the true membrane is that it should be made up of lipid bilayer plus proteins. Are you getting this? Storage bodies के आसपास membrane हो सकती है, but this is not a true membrane. Bacteria के अंदर सिर्फ एक ही true membrane है, that is plasma membrane. These storage bodies, they are known as cytoplasmic inclusions. Cytoplasmic inclusions. So I am going to write the name of the few storage bodies here. First one, it is PHB. Write the full form. Polyhydroxybutyrate. Yes, this represents reserve food in the form of fat. This is reserve food in the form of the fat. And nowadays, this polyhydroxybutyrate, it is utilized for the formation of biodegradable plastic. Yes? So what is the full form for? PHP. Polyhydroxybutyrate. What is the importance? Utilized for making biodegradable plastic. Next one. Glycogen. This is reserve food in the form of the carbohydrate, right? CH is carbohydrate. This is most common type of the reserve food present in the bacteria, glycogen. Along with that, there is presence of volatilin granules. Volatilin granules, they possess phosphate, going to possess phosphorus, right? So, this is about the bacterial cell structure. In the bacterial cell structure, here we have discussed few points. But now here I have to deal with this part of the bacteria in detail. What is this? Nucleoid. And this part of the bacteria in detail, this is cell wall. And one more structure, that is flagella. Okay? So here I will be dealing with the prokaryotic nucleoid and compare it with the nucleus of eukaryotes. So you please divide the page into the two columns. On one side you write prokaryotes and on the another one eukaryotes. In the case of the prokaryotes, there is presence of primitive nucleus, that is nucleoid, and in the case of the eukaryotes, the nucleus, well-defined nucleus is present. प्रोकैरियोट्स के अंदर ये जो न्यूक्लियोइड है, इन द न्यूक्लियोइड देयर इज प्रेजेंस ऑफ डबल स्ट्रैंडेड डीएनए. So double stranded DNA, you are going to write it down like this. dsDNA, that is double stranded DNA. This double stranded DNA which is present, that is circular. What do you mean by circular? That is two ends of the DNA, they are not free, they are joint. In the case of the eukaryotes, again one of the component which is present, that is double stranded DNA. But now this double stranded DNA, it is linear. The two ends of the DNA, they are free. Linear DNA, it is also known as fibrillar one. Right? In a human cell, yes, here I am talking about the eukaryotes. In a human cell, there is presence of 2.2 meter of DNA in each cell. हर एक सेल के अंदर 2.2 मीटर लॉन्ग डीएनए प्रेजेंट है. अब ह्यूमन सेल का तो साइज इतना छोटा है, इतने छोटे से सेल के अंदर 2.2 मीटर ऑफ डीएनए कैसे एडजस्ट होगा? सिंपल. फॉर द एडजस्टमेंट ऑफ द डीएनए देयर इज रिक्वायरमेंट ऑफ कॉइलिंग. DNA is coiled inside the cell. Yes, and in the case of the eukaryotes, coiling is assisted with the help of histone protein. So in the case of the eukaryotes, DNA is coiled. It is super coiled. It is coiled with the help of histone proteins. These histone proteins, they are basic in nature. Basic, they are going to have positive charge. In the histone protein, there is presence of 2 amino acid in rich amount. 2 amino acid इस histone protein के अंदर richly present है. These are lysine and arginine. These 2 amino acids, they are present in good amount in the histone proteins. In the case of the prokaryotes, again DNA is going to show coiling. Here again the DNA is coiled. But for the coiling, now the involvement of the histone proteins is absent. Histone protein की involvement absent है. Instead of the histone protein, there is involvement of polyamines. Polyamines, and they are rich in amino acid, that is alanine. Right? So here the coiling is there with the help of histone protein, and here coiling is assisted with the help of polyamines. In the case of eukaryotic DNA, two types of the regions can be easily distinguished, that is introns and exons. Two types of the DNA sequences can be distinguished. We have discussed in the previous class that DNA, it contains the information for the synthesis of RNA. DNA contains the coded information which results in the formation of RNA through a process, that is transcription. RNA through the translational process results in the formation of protein. It means the DNA, it contains the information for the synthesis of protein. Part of DNA which codes for protein, that part of DNA is known as exon. Part of protein in which this capacity is absent, they are known as introns. So DNA का वो पार्ट जो प्रोटीन बना सकता है, इस पार्ट को हम कहेंगे एक्सॉन. So they can code for protein. They can code for protein. That's why we can also call them as coding sequences. Right? Introns, protein formation capacity is absent. They will not form the protein. It means they do not code for protein. That's why now you can call them as non-coding sequences. So got it? Exons, coding sequences. They will code for protein. Introns, they are non-coding sequences. Protein formation capacity is absent. This is the scenario in the case of the eukaryotes. But in the case of the prokaryotes, there is only presence of exons. But here I can cite one exception. Exception is Archaebacteria. Archaebacteria, this is primitive bacteria. In this primitive bacteria, introns are present. Right? Another exception which I can cite here, that in the case of the prokaryotes, there is presence of double-stranded circular DNA. But one exceptional group, where instead of the circular, there is presence of linear DNA. That group is mycoplasma. So you got this? So these are the main differences between the nucleoid and nucleus. After this, now we will be dealing with the plasmid.